Good afternoon, everyone. My name is Alexandria Gray, and I did my presentation on mutant FUS causes DNA ligation defects to inhibit oxidative damage repair in amyotrophic lateral sclerosis. This paper was written by Hebo Wang and colleagues. I chose this paper because 2 decades ago, my grandfather passed away from amyotrophic lateral sclerosis, so I've always been interested in this disease. So what is amyotrophic lateral sclerosis? Well, it is a neurological disease that weakens muscles. So if we go over here, this would be a normal nerve cell with the axon and myelin sheath and axon terminals connected to the muscle. But if we come here, looking at a person who has ALS, with the affected nerve cell here, you can see that it is much smaller and the axon is missing some myelin sheath, and the muscle that is attached to the axon terminals is much smaller. So the muscle has wasted away. So in this paper, the ALS patients have defects in DNA nick ligation and oxidative damage repair. And so how this happens is that there are mutations in RNA DNA binding protein FUS. So these mutations can lead to reduced recruitment of XRCC1 and LIG3 to DNA strand breaks. So what they found in this paper is that a pathway was discovered of defective DNA nick ligation in FUS mutated ALS. The purpose of this paper was to provide deeper insights into DNA ligation defect in FUS associated ALS. And so they investigated mechanisms responsible for single-strand break accumulation in the neuronal genome, and they characterized the interaction of FUS with XRCC1 and LIG3. And pictured here is just a picture of a neuron. So what were the methods that were used? Well, one of the methods they used was immunoblot, which is also known as the Western blot. It is used to detect specific proteins. They also used glucose oxidase, which is used to mimic the oxidative stress. And they also use co-immunoprecipitation, which is used for studying the protein's interactions with each other. So one of the first things they did is that they knocked down FUS using shRNAs. And so what they did is the FUS shRNAs were transfected into neuroblastoma cells called the neuroblastoma SHSY5Y cell line. And they analyzed these results using immunoblot. And what they found is that there was around a 70% depletion of FUS. So if you look here, looking at the control here compared to the FUS shRNA1, there is a decrease in the presence of FUS. And it's the same thing here in the second one, comparing the control with FUS shRNA2. And the beta-actin they used was just the control. And if we go down here to B, they just wanted to quantify the results just to better show the decrease in the FUS, showing compared to the control here in the shRNA and the control here. So the next thing they found was that FUS is associated with PARP1, XRCC1, and LIG3. And so how they did this is that they analyzed these DNA damage repair proteins in both the SHSY5Y cell and also in motor neurons. And so what they did is that they isolated these proteins using the co-immunoprecipitation, and then they treated them with glucose oxidase. So looking over here, we can see this shows whether or not there was treatment of glucose oxidase or not. And so if we go over here under the minus, only the FUS is present. You can see that PARP1, LIG3, and XRCC1 are not. glucose oxidase treatment, all of them are present with or show some sort of presence with the FUS. And the results here are similar as well in the motor neuron, whereas there was no glucose oxidase treatment, and there was barely any presence of these proteins here. But if you look under the positive for glucose oxidase treatment, you can tell that there is greater presence of those proteins. So what they found was that FUS associations with PARP1, XRCC1, and LIG3 enhanced in the glucose oxidase treated cells. The next thing they found was that FUS mutations are linked to ALS. And so how they found this is that they obtained FUS mutated cultures from patients with familial ALS, and the two cultures they had were R52-1H and P52-5L. And what they did is that they analyzed the results using immunoblot, and they compared the presence of the FUS in both the nuclear extracts and cytoplasmic extracts. And so what they found here is that the P25-2L cell line had a 3-fold increase in the cytoplasmic levels here. So if you're comparing the wild type FUS with the third one over here for the P52-5L, you can tell how there is an increase in presence of the FUS. And for the nuclear extracts for P25-2L, there is actually a reduction in the nuclear level because it goes from a greater presence to it's not as great in this one. And so for the R52H cell line, there is just a slight increase in the cytoplasmic levels. So not as much as of an increase as it was in the P25-2L cell line. And you can see that here, comparing these two. And so down here in this figure, they just wanted to quantify their results as well here to better show the increase in the expression, as you can see here, and especially here in the P52-5L cell line. So in conclusion, they found that mutant FUS leads to defects in ligation and inhibits oxidized damage repair. So here you can see that if there is a DNA single-strand break, PARP1 is going to be able to recruit XRCC1 and LIG3 when FUS is present. And so that would lead to efficient single-strand break repair and then lead to neuronal cell survival. Whereas here in ALS neurons, if the FUS is mutated, PARP1 is not going to be able to efficiently recruit XRCC1 and LIG3. So that would lead to defective single-strand break repair, which could then lead to double-strand breaks, which then could also lead to neuronal cell death. So in the future, more research is going to be needed for a mechanism approach to